fairly routinely, but it costs you a lot of energy to manage the crop that carefully. And so even though you can get 2,000 gallons, the net energy return isn't that great. We'll talk about how I want to close that gap. But I think with a few modifications, and we'll talk about the three main research areas where we have to have advances to get to that 5,000 gallon per acre per year metric. And I think it's attainable. But the first thing you do is you start with feedstocks that are going to likely be legitimate. And in our case, we think that algae fit that bill perfectly. We also are highly enamored with algae because many of them are extremely productive in saltwater systems. And we think freshwater is going to be even a more limited resource going forward. And then if we can put salt water into our biofuel production matrix, that we are really going to have the ability to change how we think about deploying these systems. And if you look at the evolution of agricultural crops with time, if you look at the ancestors to contemporary corn, what we find in the wild, and right now what we work with are algae that are almost basically all this wild type. Very little cultivation or commercialization. Expertise or experimentation or evolution has gone into cultivating these for biofuel production. But you can see the transformation into what we use as a contemporary crop now, either through genetic engineering or strain selection and evolution with time for certain purposes. And I think with algae, we are definitely at this point here. And now the goal is to try to make this transformation into this commercial crop. And so I'm going to go into all the things that we look at that explore to make this happen. And the first thing we look at is a diversity of fuels that we can get out of algae. Out of algae, we can get hydrogen gas, we can get starch, which is almost very indistinguishable from higher plant starch, and we can get these oils. And the oils are really probably the main thrust of most research groups, but these starches, I think, are actually going to be very intriguing, especially if you want to feed human beings, because the glucose is very similar to the glucose we get from higher plant starch, and about 2/3 of all human calories are coming from starches and glucoses. So I think there's a real opportunity there to now think about salt water as a way to start to feed people if we have terrestrial ecosystem degradations. The other issue that we're going to look at in here is how to get more productivity out of these systems. And there's three primary areas where we're trying to get increases in productivity. One is the trafficking of CO2. And you have an expert within the department on the whole carbon concentrating mechanisms and different ways to traffic CO2 into algal cells. But most of these organisms, if we want to get to the biomass productivities that are targeted, there's actually not enough CO2 in the atmosphere to get the highest rates of productivity. And so we have to supply CO2. We have to give them bicarbonate, we have to use flue gases from other power plants potentially. But to get the maximum productivity, we actually have to increase the level of CO2, 0.04%-ish approximately in the atmosphere, but it's less soluble in water. So that's a disadvantage of algae, is they don't have as readily access to the CO2 as the higher plants do. And so when the CO2 comes in, it's usually transferred within the cell as bicarbonate. We can do this outside of the cell, convert CO2 to bicarb, transport it into the cell. But the enzyme that fixes the CO2, the Rubisco, is going to require this bicarbonate to be converted back to CO2 for fixation. So we have a case here where we have the CO2 coming in many instances we make a bicarb, we traffic this bicarb through the cell, but when it's time to fix CO2, we need to convert it back to CO2. And a lot of studies that have recently come out of places like Iowa State University, Martin Spaulding's group, is that by increasing and overexpressing these bicarb transporters, and in some cases carbonic anhydrases, you can increase biomass twofold. Now remember, right now we're about half of what the DOE wants. And what the DOE wants is about 1/4 of the theoretical maximum gain we can get. So when we start making twofold gains in this, we're taking very big steps forward in terms of productivities. The next really big issue, which I'm going to talk about towards the end of the talk, is the inefficiency in light absorption. So we have an inefficiency in photosynthesis in terms of the energetics, but we also have an inefficiency in that organisms are greedy. Their solar collectors are too big. We have to trim their solar collectors down so each organism doesn't eat as much and get full while the rest starve. We have to equally distribute that light more readily through the system. And then we have this carbon partitioning issue where basically once CO2 is fixed, there's one common metabolite that's the mother of all metabolites. And that's basically, we're going to come in here, we're going to make G3P out of photosynthesis, 3-phosphoglycerate. And then 3-phosphoglycerate we're going to be able to convert into all the other constituents, whether they're amino acids, whether they're lipids, or carbohydrates. And what we're probably the best at as a community right now is figuring out how those metabolites get trafficked so that we make metabolites that we want versus metabolites the cell wants to make, which is primarily more protein for division. So within this industry, there's been great support over the years from primarily the military, but also a lot of the aviation sector, all of which wants to get into a more sustainable space. So Boeing and FedEx are big sponsors of a number of research projects in here as they think they have to come up with aviation fuel that's a more stable price point, as well as more environmentally benign in terms of the planet. And the military has for a very long time been willing to pay a strong premium for algal biofuels. Congress is debating the intelligence of that or whether or not they should do it. Right now the military pays on the order of $300 a gallon for algal biofuel. Many members of Congress think they shouldn't pay $300 a gallon for what should cost $3 a gallon. But what the military says is it just wants to test the stuff out and wants to identify problems and see if it's a promising research approach. So they're currently paying a premium for the fuels. They have a jet called the Green Hornet, which runs on a variety of biofuels. And when they burn the biodiesel from algae, they say the performance of the jet is indistinguishable. But interestingly, they found that the biofuels were much cleaner when they were combusted, and they didn't lead to a sooting. In the military, that's a big deal because the soot means heat, and heat means better exposure from other interrogation methods. And so they really like it from a maintenance standpoint. They think it gives their aircraft a lower heat signature. They can also run their various boats and ships on the algal diesel fuels. And it turns out that when they run the drones, that the drones like to loiter. They have a very different combustion profile. It's not like running flat out. And so they have a very sort of boutique fuel composition that's optimal for them. And it turns out the biofuels are highly amenable for converting that. And I think I'll show you some examples of how we can shunt this metabolism very precisely into molecules that we want. And some very interesting molecules to change the kinds of fuels that come out. So the take home in the whole first section is photosynthesis has got some inherent inefficiencies. If we, however, optimize the whole process, we can get to a place where we can probably make this work. None of it's going to be easy, but we can actually have a legitimate production platform. Algae are probably among the most productive photosynthetic organisms we know of, almost an order of magnitude better than the terrestrial plants, and they'll grow in salt water. The three big areas for improvement are: one, reduce the antenna size so photosynthetic efficiencies increase, increase the carbon trafficking to the cell so we can fix it more readily, and then partition the fuel into products that we want that are good fuels versus biomass that is not good fuels. So we always think about this from an engineering perspective. We basically only have 4 pipes to worry about. We're going to have a carbohydrate pipe, we're going to have a protein pipe, which is cell division, we're going to have a lipid biosynthesis pipe. Those are all going to be our outputs. We're going to have photosynthesis, which is going to be CO2 fixed into our 3-phosphoglycerate, and that is going to be our input pipe. Our goals as metabolic engineers, for the most part, has been focused on expanding this pipe, this lipid pipe, as wide as it can go, so all the flux goes in there. Never, ever, ever tinker with that pipe. That pipe's got to be as open, flat out as we can possibly get it. The big enemy is cell division. Algae love just to make more of themselves, go forth and propagate. We want to stop all that protein accumulation and put it into products that we're after. The carbohydrate property, we're fairly agnostic about it. It can either be big or small. If it's small, we're able to put things into the lipid biosynthetic pathway. But the real enemy in here is cell division. What we want is a leaf. The leaf has perfected this. It emerges in the spring, it grows really, really fast, and it stops. The rest of the year, it harvests light, it oxidizes water, it fixes CO2, very challenging reactions. It makes sugar, and it gives the sugar away to the tree for the greater good. At the end of the year, the tree throws away the leaf. We want to take these cells and make them artificial leaves now. We want to take these things, do all the chemistry, do all the catalysis, give us the fuel, and make the harvesting as simple as possible. Don't grow and grow and grow and grow and grow. Self-repair, fix your catalysts, recognize when your catalysts have decayed, fix them, and stay operating, but but don't just keep getting a huge leaf at the expense of the tree. So the humans are going to be the tree, give me all that photosynthane, and then we'll discard the algae when they've served their purpose. So our first attempt at this was, okay, let's see how good we're at this. And so we went out and we knocked out carbohydrate biosynthesis. And so in our laboratory, we have a fairly stock set of experiments that we do. We take algae that we have genetic manipulation tools for, we make a whole library of mutants, and we screen those mutants for phenotypes of interest. It's a very classic industrial microbiology approach. You know your phenotype, make mutants, screen for it. And so in the laboratory, we look for some organisms that will make hydrogen gas. It turns out, and I'll talk about this a little bit later, some of these organisms, when they have a low potential electron and have nothing else to do with it, they dump it into protons and hydrogen gas comes out. And some of these algae, rather than evolving oxygen, will bubble hydrogen gas like a witch's cauldron for about 2 weeks. And then it will stop. And so we developed a screen with this using palladium and tungsten oxide. When the hydrogen reduces the tungsten oxide, it turns blue. We can put this sensor over our algae on agar plates. And if they make hydrogen, it will turn blue. And this is a mutant that doesn't make any hydrogen. We can use fluorescence to look for the oil droplets. We can use iodine vapor to look for starch. These are starchless mutants that we found. And we can use fluorescence to look at the state of the photosynthetic electron transport chain. If the photosynthetic electron transport chain is backed up, for the most part, your fluorescence increases, and you can monitor fluorescent efficiencies that way. So the first case study we did is we looked at these starchless mutants. This is the wild type up here. These are 2 different mutants that we found that didn't make any starch. So we found 2 different genes, the Chlamydomonas, reinhardtii, green alga, that don't make any starch. The hypothesis was if we block starch, that pipe should shut down, our lipid pipe should open up, and we should make more oil in these because they can't put that photosynthate into carbohydrate. The other thing that we always have in algae, and anybody that studies algae, they always do this, that there's a plus nitrogen and a minus nitrogen experiment. The minus nitrogen is always done to stop protein biosynthesis. So that's one of the other pipes is protein biosynthesis and cell division. One great way to stop protein synthesis is to take away nitrogen or take away sulfur. Those are very common techniques. And then you totally shut down that pipe, the protein pipe. And what definitely happens when you do that is your pipes to carbohydrates and your pipes to lipid. Those open up and you take more flux in those directions. So now we're going to go in there and knock out the carbohydrate pipe, and we can use minus nitrogen to knock out the protein pipe, and we should get the mother load of lipid, right? This should be just a really home run, easy slam dunk experiment. And so as you take away nutrients, you get this huge increase in carbon stores, whether that be oil or starch in the wild type. And now the mutant, the question was, what would the cell do if we knocked the other starch out of the biosynthetic pathways? And one of our mutants was looking like it was trying to make a little more oil than the wild type, and so we were fairly excited about that, and we kept pushing this. And we could let this strain grow for a couple of weeks, and this would basically then become a bag full of oil at a certain point. I mean, there's tons. The whole cell biomass is basically oil. It's about a 60 to 70% oil cell, such that when we centrifuge it, the cells go to the top rather than the bottom. But then we started to check under the hood on this more carefully. We did a couple of other things on this. The first thing we did is to do due diligence. We put the gene back into the organism that made it starchless. And that transformed cell went back and it made starch again. There's our mutant, that's just the iodine by itself. And when we put that gene back in the organism, something very strange happened. The students, every time they spun it down, these are the transform lines down here, thought their cells were contaminated because they had a big white film over the top. Now in this particular organism,